smell is more important for the animals, for their sexuality, for their survival, for their nutrition, for their protection, defense, anything, okay? But it's not the case with humans, okay? Our emotions are affected when we are using good smells, perfumes and all that. Definitely, it gives us a pleasure and it changes our psyche to some extent. But it's nothing like that. These are causing a significant effect, a significant change in us, okay? Bad smell, good smell does affect us, okay? And sometimes we have some responses or reactions also for the bad smells, you know. Some bad smells are so bad that the person can start vomiting in response to that. And we know that if there's a certain bad smell coming and you cannot tolerate that, the person will throw up, will vomit, okay? So these responses do happen in humans, but it's not really significantly affecting us so much. So in our brain where we perceive the smell, we have different areas. Qadim area for the smell, for our Qadim purpose really. Some emotional responses to smell, and for some primitive reflexes. For example, when you are hungry, alright, and you smell something, the response is that all of a sudden there is some salivation and there is fluid coming in your mouth. Okay? Your mouth is full of water. Okay? These kinds of reflexes in response to olfaction, they happen in human beings also. Okay? These are old primitive reflexes. You are not doing it on purpose, it happens automatically. So, old reflexes and some emotional changes, they are mawjood. But in humans, they are not so important really. So this area should be very little area in our brain, which is represented. Okay? We have a very, very old area, Qadim area, very Qadim. The other area is Qadim, but less Qadim really. And one area is Jadid area for the control of the smell or for the understanding of the smell. In humans, this Jadid area is more important because by this Jadid area we can differentiate different smells and we can characterize different smells. Now you know you're using one perfume, you're switching to another perfume, and you are smelling other perfume also or any other odorant substance and you can easily discriminate and you can understand that substance pretty well, and you can have in your memory that substance, good memory or bad memory about that, and you can compare. So you will prefer that substance or you will avoid that substance, and you know it was related to something bad experience, you will never use it again. So these are the characteristics which we use where we can think and discriminate. This is a Jadid area in our brain. So we have got different areas in the brain for understanding olfaction. Qadim area also mawjood in animals. We don't rely on that area much, some reflexes only. Some areas which are less Qadim areas, I'll talk about that also, and Jadid area is another area, okay? Okay, so let's get started very quickly because it's a very simple sense. Okay, you just have to follow what is happening. No difficult mechanisms, nothing very special. You need to know few things about smell. Well, there are so many different kinds of smells that we can appreciate and we can perceive, but it's difficult to describe all really. You understand the difference, but describable difference is not there really. You can describe few only, a range. Sometimes we classify the smell as the primary smells. There are some primary smells which combine together, making combinations, and then you develop hundreds of different types of smell by combining different smells. Good smells and bad smells, chemical smells and non-chemical smells. When you combine them, you come and discover a new smell. You are mixing two substances, the smell will change really, isn't it? So it's a combination. So there are some primary odors we regard, but there is no fixed classification. There are 6-7 smells. I'll give you few names like that. One smell is a floral smell, like of Zuhur, you'll say the flowers, isn't it? Different flowers. You call generally the floral smell. A pleasant smell, definitely. Okay? One smell is like Peppermint, the mint smell, the smell of nana, okay, typical. Pepperminty or mint smell, that is another basic smell we consider. One smell is a very pungent smell which stimulates you and you are irritated by that. You are working in the lab, chemia over there, and you are making ammonia gas or HCl gas. If it goes in, the fuel goes inside, you are so much irritated, it goes right into your brain, really irritating the whole passage. That is strong and pungent smell, okay, that is one variety, a chemical type of variety. One smell is camphor. You understand camphor? Kafur. I don't know if you understand it or not. A typical smell. Okay? Camphoraceous smell. It is one of the types of smells. Okay? And in this way there is one putrid smell. Putrid means something which is rotten. Okay? Decaying animals. If an animal is dead, a dog or a horse, and you are passing by, the smell which comes in your system, you just avoid that. Sometimes you vomit from that smell if it is very strong. So putrid smell or a bad smell, a decaying smell. Okay? And these are the different varieties. Ethereal smell, ether smell, chemical smell in the lab. So these are taken as the primary smells. When we combine these smells in different proportions, you develop new and new and new odors or deodorants. Okay? So there is no need to remember all these things. I'm just giving you an example that we have a variety of primary odors, and when we combine the primary, we can develop hundreds of different types, and we appreciate those smells. Anyway, olfactory membrane. Okay, you know about the membrane which is modular here. Now we are just into the pathway. We are starting the pathway of the smell, the neural pathway. We'll take up stations and we'll go to the destination in the brain also. You don't need to remember the pathway, each and every station and detail of the pathway. Just have an idea about how the pathway is going on. Okay, so the pathway starts from the receptors and the pathway finishes at the merkaz in the brain. You know that. Our starting point is the receptors. Where are the receptors located? In the nasal mucous membrane, a specialized part, upper part, olfactory membrane, and that area is supposed to contain a specialized neuronal setup. Okay, now this is your mucous membrane here. All right, this area is the mucous membrane area, and in this mucous membrane, this is the layer of mucus. So if we go into the nasal cavity, up, and touch that part really, nose, okay, we are touching the olfactory epithelium, up in the nose, towards the roof of the nose here. Okay, exactly. You have the roof of the nose, by the way, anatomy, coming back to anatomy. A very thin bone is there, a perforated bone is there, which is forming the roof. What is that bone? It's a cribriform plate of the ethmoid bone, if you remember. Okay, it's a porous plate. Openings are there. Olfactory nerve bundles pass through that. Yani from the nose, from your starting point, the olfactory nerves will be formed, and the olfactory nerve bundles will pass through that thin plate, and it will go into the brain, really, isn't it? So it means we are starting our station or our journey from here. It is the lining of the mucous membrane here, and if you touch that, if you touch your nose, it is kind of lubricating material, a little mucus is there all the time. It is not dried completely. Okay, if you just touch it, you see a clear transparent mucus is there, and that mucus is secreted by the mucus gland that is lining all over. All right, so here you have the mucus. I'm talking about the top area. When I'm talking about the top area, just this area, literally roof and medially, this is the olfactory membrane here and here. In this area, olfactory membrane is modular. If you see histologically, it is a different area, yellowish color, light in color. Okay. Anyway, looking into that area, you have the neurons module. Look at the bipolar neurons which are situated. Typical shape. Bipolar means two poles. One pole is module in the mucus side towards the cavity. The other pole would be going, the afferent fiber is going, and that afferent fiber is collecting and forming the olfactory nerve bundles. Okay? So a bipolar neuron, no synapse, it's a neuron itself. One end of the neuron is having the receptors. The other end of the neuron is forming the afferent nerve fiber, and that afferent nerve fiber will give you olfactory nerve bundles. Olfactory nerve bundles will pass through the cribriform plate of the ethmoid bone, and then they will enter into the brain. Okay, let us get back and look at what is happening here. The mucus glands are there, supporting cells are there, these are the neurons, your neurons, and the other end is divided into olfactory cilia. Okay, the other end of the bipolar cell process, it divides into branches, and these branches behave like cilia. Okay, the cell membrane is modular and finger-like projections are there. They are modular in the mucus. Now these processes have the receptors, and anything which you are smelling is going into your system, and that thing has to get attached to the olfactory hair, these olfactory cilia or olfactory hair. Once they get attached, then there is a reaction going on, a triggering, and the pathway is stimulated. We'll see how. Any substance you are smelling, for example, I have a substance, anything which is placed here in a cup, okay, that is a substance, and I want to smell that substance. What should I do? Okay, one thing I'll do, I'll bring that cup and I'll sniff. I'll have typical sniffing, okay, so that, okay, is that substance really, is the liquid going inside me? Okay, you know, by sniffing, the liquid will start going inside my nose. It will not happen. This is not the power of sniffing, you know that, okay? So still I can smell, and why? Or if substance is very strong, it is placed here, and still I can smell that substance from here, from that distance really. Why? Why am I smelling that substance? Particles. Okay, so what is happening to the particles? Okay, they are moving up in the air, they are moving into my system, they are going into my mucosa, they are touching the mucosa, and they are penetrating the mucosa, and they are going to the olfactory hair cells, and they are binding with the cells, and somehow they are stimulating the mechanism, isn't it? This is what we expect. So what is that process known as? What is the characteristic, what is the property which is causing it? Volatile nature. Thank you, Amir. It's volatile nature. First property or condition, the substance has to be volatile. If a substance is totally non-volatile, and if you bring it and you try to sniff it, will it give you any odor? La. You will not say la. There is no odor because the substance is not volatile. The more volatile the substance, the better it moves into your system. So the first condition is volatile nature, volatility or volatileness of the substance. That is the first thing. After having this volatile characteristic and it is going into your system, reaching your system up, what should be the second characteristic in order to stimulate? And to stimulate your system, what should be the second characteristic, physical characteristic? It has to go into the mucus now, the mucus which is modular, okay? Volatile means it is going from here and it is going and it is reaching here. Khalas, it is volatile. Perfect. Now it has to go here. What characteristic do you want? Diffusible. If you are saying diffusible, what characteristics would you like to have in diffusibility? Good. Mothers, yes. It should be soluble. Now you are saying soluble. What? Okay, hold on. Okay, don't accelerate. Solubility, okay, lipid solubility, I take the answer. What is your choice? You are passing through the mucus. Would you like to have water solubility here or lipid solubility here? In the mucus. First you are in the mucus, okay, which is secretion, which has carbohydrates, which has proteins, which has water also. So what would you like to have in the mucus? Now after crossing the mucus, yes, the water solubility. Now you are crossing and now you are touching the cell membrane of the olfactory hair. What property here would you like to have? Lipid solubility. Yani you need three properties. Awal, volatile. Number two, to some extent it should be water soluble, and to some extent it should be lipid soluble. That will give you a perfect odorant substance, okay? Different odors have different characteristics. That's why some go fast, some go slow, some go more, and some go less. It happens like that. Are you getting the three properties? So these are the basics for taking them to the receptors. Alright? Now, we talk about receptors. This is your nose portion here. Okay. These are the olfactory nerve bundles which are passing. They are passing through a bony plate. My hand is the bony plate here, and this bony plate is the cribriform or the ethmoid bone. Okay. Through this the bundles are passing. It means the holes are modules here, and through these holes hundreds and hundreds and hundreds of bundles are passing. It's not one olfactory nerve. These are the olfactory nerve bundles, small bundles which are passing up. Is that clear? Now, you are into the brain. Yani, you have entered the brain, the cranial fossa. You are in the cranial fossa now. And you are directly entering into an area of the brain because these are the bundles which are passing and crossing, and after crossing, they are entering into the brain. Now, this is the inferior surface of the brain. Now, my two hands are the two cerebral hemispheres. Frontal lobe and the occipital lobe. Frontal facing you. Alright? Here, the fibers are coming and they are directly entering into which part? The frontal part or the parietal or the occipital or temporal? What do you think? Frontal. They are right entering into the nose. Above the roof of the nose they are going. So they are reaching the inferior surface of which lobe? Definitely the frontal lobe. There is no other choice. Okay. Over the inferior surface of the frontal lobe, there is a swelling or a bulb-like structure, bulb, olfactory bulb, which is there. Okay. So their destination is to directly go into the olfactory bulb and look at the picture of the olfactory bulb where the olfactory nerve bundles are going. Can you see that? Olfactory bulb is there, a bulb-like structure. Is it there? This is cut section, basically a cut section. Okay. And this is the bulb. This bulb is going back like this. This is the swelling part. And in the olfactory bulb, you have some special histological structures. Can you see the rounded structures made up of the fibers? Okay, yeah, like rounded, a glomerulus-like thing. Okay, spherical areas, small spherical areas, which are made up of the intermingled fibers. The fibers are synapsing and you are making a ball-like structure, and that is one ball. That is another ball. Okay, many fibers are there and it's like that. So thousands of balls are there. These are the glomeruli, or the glomerulus is one. Okay. Now these olfactory nerve fibers, they are going into the glomerulus. They will synapse. It means there are some other cells modued in the glomerulus, or the endings of some cells are modued in the glomerulus. Okay. Well, in the glomerulus, you can see different cells are modued in the olfactory bulb, and they are sending their fibers here also. Well, they are synapsing with the, so the first synapse of the pathway, where is it happening? In the olfactory bulb, and well within the glomerulus actually, okay, in the glomerulus. It is the first pathway. Alright. The cells which are modued in the olfactory bulb, they are called the mitral cells, they are called the tufted cells, and they are called the granule cells. How many cells are there? Mitral, tufted, and granule. According to the histological characteristics, okay, they have the tufts, they have a typical shape, you call it mitral, and they are dot-like or very small granule-like cells, so they are granule cells. Okay. Well, the mitral and tufted cells, remember two cells, they are synapsing with the olfactory nerve fibers. The mitral cells and the tufted cells, they are synapsing with the olfactory fibers directly. Granule cells are not. Okay. Granule cells are for some other purpose. They are the regulatory cells or the feedback cells. They are not the main cells of the pathway, please. Okay. So two cells in line, mitral and tufted, they are synapsing. Halas. Okay. Now, mitral cells are shown here. Tufted cells are also there. They are also sending it. Their fibers are collecting and the fibers are going back now, and the fibers are forming the bundle. So the second order neurons in the pathway are mitral cells and tufted cells. Their axons will pass posteriorly, backside. Okay, these are the two hemispheres. On the inferior surface, a bulb is modued and a bulb is modued. From the bulb, there is a tract, a longitudinal tract going in toll actually. Okay, this tract is called the olfactory tract on the inferior surface. From the bulb, the tract is going back. That tract is made up of the axons of the two cells mainly. What are these two cells? Mitral and Also the granule cell fibers are coming, I'm not saying that, but the main cells which are forming, these are the, alright? Okay. So olfactory tract is formed. Olfactory tract will go back, and after some distance, this olfactory tract, now this is olfactory tract coming like this, and this olfactory tract when going back on the inferior surface, now it will break into two parts. One part is going medially and one part is going, okay? This olfactory tract divides into two main strips, medial and lateral. The medial one is called the medial olfactory stria, the bundle. The lateral is called the lateral olfactory stria, very simple. There is a very fine middle bundle also coming, a middle thing is coming, and that is also going to some area of the brain. But there are two main bundles. One is medial and another is there. Is it okay so far? Alright. Look at the diagram again, the same thing in a side view again. You are looking here, bipolar cells, this process here, the receptors are module olfactory here, typical cell body, afferent fibers, these are forming the olfactory. This is the cribriform plate of the ethmoid bone, do you see that? Through that opening, it is passing and it is entering into the bulb which is module here. In the bulb you have got mitral cells and the tufted cells synapsing, and they are forming the olfactory tract. The tract will go back and it will divide into two parts. One is medial, other is lateral. Okay? Alright. After going medial and lateral olfactory stria, okay, these stria will pass to different areas of the brain. Okay? Medial olfactory stria pass to the most qadim areas of the brain, okay, for the smell. The lateral olfactory stria will pass to less qadim areas, and a little middle band, I told you about, a very fine band in the middle, that passes to a jadid area, okay, through the thalamus, and it reaches the cortex, the frontal cortex. Well, smell is considered to be wahid sense which is not actually passing through the thalamus. All other senses are passing. This is what we say. But the latest research says that a little part of smell still passes through the thalamus. It is the jadid component of the smell which is passing through the thalamus, synapsing in the thalamus, and then reaching the highest station which is your cortex. Okay. So, but by and large, we consider that we see the medial olfactory stria and the lateral olfactory stria, they are not passing through the thalamus, they are directly going to the brain areas, qadim area or less qadim areas, but still they are managing without thalamus. Okay? Anyway, mitral tufted cells, they are going out, the same diagram really, nothing special about that. Now, what are the concepts or the areas? Basically, I'll talk about the very old system, and the new system that I already talked about in the beginning, if you remember. Let's have a few words more about these areas. Okay. So, what happens is that the very old system is a primitive system, also module in the animals, and they rely on that system for some reflexes of the smell or some changes in the behavior and emotion in response to it. Okay. So emotional and behavioral changes in smell in us, if it's causing a sense of pleasure or a bad, or we are having some change in the behavior according to the smell, then that is done by the very old system. Okay, the very old system is controlling that. The old system is controlling some areas about the smell understanding, where we develop our experiences, our learning about the smell from our childhood. Remember you yourself in the childhood, when you were a child and your mom used to give you different things, trying different things, foods, because you are growing up, and moms always want children to eat everything really, okay? Whatever they cook, they would like to eat, they don't want a particular choice that you will only eat batatas or you will only eat one thing, because kids usually want one thing which they love, okay? But moms, they know that for growing up, everything is needed. So they would like to give you some khadar also. We never liked those in childhood really, okay? Keep it away, all those things, okay? Give something which is pleasurable. So anyway, and once a mom is trying, you're very young and she's trying something new, okay? And the first spoon goes into your system and it is a little bitter or it is having more salt or something, like this, and the child do in such a way really, such a bad experience. Well, that experience will be remembered by the child, it is saved in the memory. Second time when the mom tries, she fails simply, so she withdraws. Any other time in your life when you see the same thing, if you remember, or if you taste the same thing, no, I will not have it, definitely, okay? Even though it's not bad, but since you have developed a very bad experience with that, that is saved. So those discriminations which develop, you're learning about the foods, your likes, your dislikes about the food. You like something, you dislike something for certain reasons, for certain experiences. That conditioning or that learning is through the second area, the old area. So the old area is controlling that part, okay? Your experience is about smell and related to the food and other things, okay? Your behavior, emotions and your some reflexes spontaneously by the qadim, very qadim area, very old area. Are you getting the point? Okay. So smelling bad food, something like that, and having experience saved and you avoid that thing, that is coming through the old areas. Alright? And new areas, when you can distinguish the smell and you select few things by consciously, a conscious selection of different smells, different foods on your thinking, on your basis when you are grown up. So you understand, your understanding is increased. That is done by the Jadid area. Okay? Look at the areas. Very old olfactory area, it's around hypothalamus. Some old areas of hypothalamus, they are important for that. They control the very basic or the very old attributes of the smell. Okay? Less old, which are old but not very old, they are controlled by some other areas of your limbic system also, like it's amygdaloid body, amygdala or prepiriform and piriform cortex, and some areas of hippocampus and the limbic system parts. They are controlling your experiences about learning of smells and relation with the food. That is done by this less old area. The newer pathway, Jadid one I said, okay, that is passing probably through the thalamus, and from the thalamus, it is passing indirectly to the orbitofrontal cortex. Okay, so this is the only part. The new part is the only part which is passing through the thalamus probably. Other parts are not passing through the thalamus. Is that clear? This is about the pathway. So you started the pathway again from the receptor. Receptors are module. Actually, receptors are nothing special. I am getting back to the receptor. Look at the story again, please. I am receiving a volatile substance in my nose. It is going into my mucus. Mucus dissolves it because it is water soluble. Then it touches, now, before that in the mucus, a very interesting thing. In the mucus, there are certain proteins module, very special proteins, which are there for the istiqbal of the substance. For example, alright, you are the substance, you are coming, okay, and I am the protein, okay. I am just wandering here and looking for you. You come, oh, you are there, come on. And I say, okay, come with me, come. And I take you and I to the membrane in this way, okay. So, the protein will catch, bind. It is called the odorant binding protein, okay? And it will take it along and both will go and I'll fix it to the receptor which is module here. And thank you very much. Okay? Have a seat, please. So you got the point. Odorant binding proteins are there to carry that in the mucus so that, because he is Jadid here, he doesn't know where to go. So Khalas, no problem, come with me. I'll take you. You are here, that's it. So there are different types of odorant binding proteins which are there and different substances are coming. They select, like attach, and they take, and they bind, and khalas. This is how they communicate. You got it? So first mucus, odorant binding protein, and then attaching to the olfactory hair membrane where the receptors are module now. Now when you stimulate the receptor, what will happen now? A mechanism, cyclic AMP or some mechanism will be stimulated. G protein mechanisms are modular in the cell membrane. Okay, they will be stimulated. Usually the G protein coupled. Opening of the receptors channels, and these channels are the sodium channels or some other ion channels. If they open, what will happen? Sodium will come inside. When sodium come inside the bipolar neurons, what will happen to that neuron?